That's okay. So I'd like to start out by telling Andy Ewing congratulations. Very good talk, and congratulations on receiving this well-deserved award. I'd also like to say exactly the same thing to Bo. Congratulations to both of you for an excellent and fine job. If I have my thing working. Yep. So, the opportunity like this is too good to pass up, to make fun of Andy especially, okay? So I just had to do this. So I was looking through my pictures, and they go way back. I mean, we can get just about anything. But one that really caught my eye was this one. Okay, let's see this one. Do it. This one. This is a picture of Andy that was taken in about 1982, I would guess, 1983, something like that. And in case you're wondering which one is Andy, it's the handsome one right there, okay? And what's really interesting, I think, about this picture is if you just zoom in on Andy's face, you zoom in on Andy's face, you can see that hungry look, like, he was going to outride all of us, just like he's done really good science over all the years. He was going to ride really hard and charge forward. So we all have aged a little bit since that time. I think you can definitely tell that's Andy. Okay, what I wanted to do in my talk today is tell you about some recent activity that's been going on in our lab in this area of fast-scan cyclic voltammetry that Andy was nice enough to introduce. What we're trying to do here is we're trying to measure dopamine neurotransmission. So there's a schematic here of the dopamine nerve terminal, the neuron that is targeting on is called a spiny neuron or spiny cell, and then there's glutamatergic input there as well. So what we're trying to do is a long-term goal in my lab is unravel the chemical events that are going on here and that are doing processing of information so that the animal can do normal behavior. But it's a really complicated issue, and this is sort of a diminished view. Notice that this is 0 to 100 microns here of the kind of complexity that exists in brain tissue. They're not just dopamine nerve terminals in these regions that we go to that do in fact have a lot of dopamine, but there's other things, blood vessels, glial cells, the target neurons that are receiving the dopaminergic input. And what we want to do is put an electrode down in the brain and fish out the chemical signals arising from dopamine fluctuations and try to understand what they're doing. And so we're sort of putting together a tool chest of things that we can use to do this. So if you were here during Andy's talk, you saw the carbon fiber microelectrode that we've been using for a very long time now, and the idea of fast-scan cyclic voltammetry. And you realize that the output of this experiment then is a cyclic voltammogram, if we're in the presence of dopamine, that tells us, yes, in fact, it is dopamine, and whose amplitude is proportional to the dopamine concentration. So just to give you an idea of how one of these experiments might go, let's picture this being a side view of a rat brain, a rat that is anesthetized. What we'll do is put down into the rat's head a stimulating electrode, and we'll place it back in the rear of the brain where the dopamine cell bodies are so that we can electrically excite them. And then we'll take our carbon fiber electrode and put it in the middle of the dopamine nerve terminal. So the idea here is if we stimulate here at the same time the stimulation occurs, the electrical action potentials will propagate up the line. They'll induce the release of dopamine here. Okay, so that's this experiment in about as simple way as you can conceive it of doing in vivo electrochemistry. And if you do such an experiment, and probably many of you have heard me speak about this before, you can very nicely see dopamine. So there's a dopamine cyclic voltammogram again. We use these color blocks like Andy described, where we have the applied ramp on this axis, the data acquisition time on this axis, and right there at that red bar, that's where the stimulation was turned on. And this peak is that peak, so the dopamine oxidation peak. If you look at what happens at the potential where dopamine is oxidized, you see the signal go up during the stimulation. You can also see out here at later times there's something else, and it turns out this is due to changes in pH. We can follow that as a function of time by taking a cut here. And you can see it doesn't change during the time of the stimulation. It does at later times. And we could correct this data for this data and end up with a signal that tells us about what dopamine does. So we've been doing this kind of experiment for quite some time. The goal now is to try and look at spontaneous dopamine release events while an animal is doing behavior. And I'm going to give you some examples of that, but what I'm primarily emphasizing here is the way that we've gone about developing the software to do this. So we were recently fortunate enough to get a grant from NIH to hire a programmer to really upgrade the software that we use for these kinds of experiments. And one part of the upgrade was we control the potential and handle all the functions that are needed using National Instrument cards. Used to be it took 3 or perhaps 2 cards to do what we want to do. Well, these cards have got better and better, and the programmer figured out so we could just use one card. And the card does things like generate those triangular waves that I'm showing you, collects the current data in the form of a voltage, and then allows us to plot it in any way that we want to. As you will see, digital inputs are also important in these kinds of experiments, especially with behaving animals. So this very same board lets us control everything that we need to control during behavioral experiments. So let me just talk you through this program a little bit and show you what various screens look like in the program, and then give you some examples of what we've been able to do with these sorts of things. If you were an operator in the lab collecting data in the rat brain, this would be the sort of thing that you would see on the screen. Basically, down here are 3 plots. One, the color plot that Andy described and that I showed you. Here is the current versus time curve. In other words, where you set this cursor, it tells what concentration changes happen there. And then this cursor reads out as this cyclic voltammogram, so you can see exactly what it is you're recording. The plot that you see up here is actually like a movie or a TV screen of actually what's going on in lifetime, and I'll show you a little bit more about that in a minute. But if you look, there are various buttons on here. One of them, one is very important, says set up experiment. So what you would do if you push that button, you go to yet another screen, and this screen would allow you to select the waveforms that you were going to apply to the working electrode or electrodes. Down here you can see an oscilloscope trace of this program, and it's actually generating 4 different waveforms. You can apply 4 different waveforms to 4 different electrodes and collect from all those electrodes if you so wish. You can build these waveforms in a piecemeal fashion, so you just select little portions and draw them out on your screen here. You can select various waveforms that are already in LabVIEW programs, or you can select a file that you built in Excel that would be the waveform you're going to apply to the electrode. So a lot of flexibility here in being able to do this. This live color plot that I mentioned, that you get to see all the time, you can select which thing you want to look at. Do you want to watch how the current at a particular potential changes as a function of time? Do you want to have the oscilloscope function so you can look at the applied waveform and what the current is as a function of time that's going on during your waveform? Or do you want to look at this color plot, or perhaps you want to look at the digital inputs and outputs? What's been particularly interesting to us is this ability to look at color plots in real time, because it really lets you see exactly what's going on in the region of the brain where you have your electrode. So in this particular waveform, we're going out to a very negative potential where we can reduce oxygen. So you can see fluctuations in oxygen concentration here. At 120 seconds, we turn on the stimulator, and that peak are the two peaks for dopamine, and there is a pH-induced change, and then you see a big oxygen change here after the stimulation, followed by another big oxygen change right here. And these are things that we've studied before and I've talked about at Pittcon, but the ability to watch them going on as you do the experiment, either the spontaneous events, which are really exciting and interesting in freely moving animals, or to see exactly what kind of response you're getting in an anesthetized animal, is a capability that we never had before. And of course, modern day computers are so powerful they can handle this large amount of data. We have, in the past, when we started, in fact, Adrian Michael wrote one of the original programs for this, we would do just a single run and then we would scratch our head and say, oh, what should we do now? And then I think it was Jill Venton figured if we did multiple runs like this, then we could signal average them together and get much improved signal-to-noise ratio. So most programs that exist today allow you to get data in this mode or this mode. But clearly the way you want to be able to do it is to collect all the data all the time, and then during the analysis of these data go back and select the parts of the data where important events happen, throw away the rest of the stuff. And so because, again, modern computers can store lots of data, it's very easy I shouldn't say very easy. I didn't do this, the programmer did it. But anyway, he wrote the program so we can continuously collect data for as long as you want and display up to, several minutes approaching an hour's worth of data simultaneously. As you can see in these long displays, you start having drift that sort of messes you up, but you can go back in there and fix that, and just this crude representation of the data gives you an idea of the events that are going on. So here's the voltammetric data, and then these are TTL responses down here. Let me just explain what that has to do. Well, before I show you examples of what we can do here, I need to just explain to you what we're going to look at here. So I want to show you an example where we are taking and putting our electrode right next to a dopamine nerve terminal. And as Andy told you, this dopamine's inside vesicles. And we're going to give to the rat an IV injection of this drug, Tetrabenazine. And what Tetrabenazine does is it blocks a transporter that's on this vesicle that loads the dopamine into the inside of the vesicle. When you block this transporter, then as Andy told you, the concentration is very high in here, maybe half molar. It's very low on the outside. Andy measured it to be 1 micromolar or less. So you have a concentration gradient across that one membrane, something like 5 orders of magnitude. You give tetrabenazine, now there's no pumping action to pump the dopamine into the vesicles. What you would anticipate, although this hadn't been shown before, what you'd anticipate is a massive dump out of dopamine. And if it comes out of the vesicles, perhaps through the transporter, it could get pumped back out to the outside, and we might be able to see it here. So we wanted to do this experiment. We wanted to do it for a long time, but we didn't have this capability of continuously looking at the data to follow what goes on in time. When we got this program, we could do that, and this is what we saw. Let me talk you through this rather complicated looking slide. First of all, we did an electrical stimulation right here, and we can see dopamine release, and that's how much dopamine release there was. And after the electrical stimulation, there was a pH change. Okay? Then the drug was given, and we're continuously collecting data, and we're subtracting these data, these cyclic voltammograms that you see here, from what was collected before the drug was administered. So you can see now we're way out here at around 6 minutes, much longer than we would normally go. And you can see everything we're seeing is dopamine. This dopamine is just dumping out into the extracellular space. If I just take a cyclic voltammogram along here, there's a cyclic voltammogram. Much like those I've shown you before, and much like we've seen in vivo before, except for this. The concentration here is absolutely massive. It's approaching 1 micromolar. Spontaneously, you never really see that. So they took a pharmacological agent to get to that very high concentration. With this new continuous data collection, you can see it. After you've collected data for a while, you can go back and do the electrical stimulation again. It's the exact same place, everything's the same, except the tetrabenazine is still on board. When you stimulate, there's no evidence for release whatsoever. That's the dopamine release curve. The pH change is still there, still the same magnitude that it was before, but there's no dopamine to release because the dopamine that is going to be released comes from vesicles. We just knocked all the dopamine out of the vesicles. So these pharmacological agents take time to get to the brain, as Andy was saying, and they take time to exert their action. You need these long time scales to be able to look at those kinds of changes. Okay, now I'll tell you a little bit about the TTL aspects of this. Of course, it's very easy on these National Instrument cards to get TTL signals, and we use them for all sorts of peripheral things. So they look like this, ups and downs. We have as many, I think there are 24 different lines that we can get of TTL signals. And they're used to do things like put out a signal to turn a loop injector to introduce a reagent in a flow cell experiment. So we can collect these kinds of data here. They're used to turn on an iontophoresis machine. So this machine applies a potential to these very small barrels and causes ejection of small, tiny amounts of material from these barrels on the order of femtomoles per second. And by turning those barrels on and off, we can, with the electrode, monitor electroactive substances, and we can have very precise timing on that sort of thing. So an experiment that I have talked about at Pittcon before, but I still find very intriguing and I think is going to lead us to a lot of insights into what's going on, is called intracranial self-stimulation. And what this experiment is, you train an animal to press a lever to deliver that electrical stimulation I've been showing you to itself. You can train an animal within about 20 minutes to do this by pressing a lever. He gets really excited and really likes to do this. So then after he's learned to press the lever, we make the task a little more complicated. We have a cue, and this cue is in the form of a light and a tone that come on. And then 2 seconds later, there's a lever that pops out, and then when he goes and presses the lever retracts, the tone and the light go off, and a stimulation is delivered to him of exactly the type I've shown you before. So, this is part of our goal to try and figure out what does dopamine do. You probably all know from Newsweek and magazines like that that dopamine makes you feel good. But is that really true? I mean, they didn't have in vivo voltammetry to be able to make those kinds of decisions. And what our research has shown is that it doesn't necessarily make you feel good. We do see dopamine release here when the electrical stimulation is, but that's because we force it to occur. What we find is that with time, the dopamine release, the spontaneous dopamine release, occurs at the cue. And let me show you what I mean by this. This is a video clip of this experiment. And the cue is going to come on at 2 seconds right there. You see that dopamine that just spontaneously appeared. This dopamine appeared because the animal went to the lever and pressed the lever. And I can show it to you one more time. See why we need to do these fast scans, as it all happens really quickly. There he is going to the lever, there he presses the lever, and there he gets the electrical stimulation. So to be able to record all this, of course, you've got to record all the cyclic voltammograms, but to get these signals synchronized with the behavior, you have to have good records, very accurate records of the TTLs and when things happen. So that's why they're so important to us. Another thing that we've been adding to our toolbox is iontophoresis. So the way you make an iontophoresis probe is diagrammed here. You take 4 pieces or a 4-barrel set of pipettes. You put into one of these barrels a glass tube in which there's a carbon fiber. And then using a conventional capillary puller, you pull this down to a fine tip. And your end product then is a carbon fiber microelectrode that's insulated with glass, and which has these barrels here which we can use to introduce drugs. And we introduce the drugs by applying an electric field to these barrels, and material gets ejected out by the combined processes of electroosmosis and migration. So if we just have a neutral molecule, it's just going to be electroosmosis that carries the molecule out. Now, people in the physiology world have used iontophoresis since the 60s. The problem is they did not have the cyclic voltammetry barrel attached to it, so they never knew how much they were ejecting. And so if they were trying to eject a physiological amount, they wouldn't know if they had or not. They just looked to see if the cell responded. Well, the cell might not respond because it didn't have the receptor for the substance that you introduce. So it was a very sort of circular set of arguments. With that carbon fiber alongside, we can, of course, do cyclic voltammetry while we make the ejection. So if we filled the barrel, say, with dopamine, and this is an experiment done in a beaker, we could eject out dopamine, and we could follow it by cyclic voltammetry, and we'd know that when we turned the ejection on and when we turned it off, things went on and off exactly like they were supposed to. But say you have a drug that's not electroactive, and unfortunately, a large majority are electroinactive. You do one of these ejections, this is what you see, absolutely nothing. So this raclopride that I'm showing you is a drug which interacts with dopamine receptors and blocks a certain class of dopamine receptors. Very important for us to understand what these classes of receptors are doing, and so we need to be able to have these dopamine receptor blockers. We need to know how much we're putting out. So what we decided to do some years ago was to combine the injection of this electro-inactive species with acetaminophen. So the acetaminophen, structure shown here, Tylenol as you know it, it is electroactive, and yet it has no physiological function in brain tissue. And we've tested this by looking at stimulated release of dopamine with and without the presence of acetaminophen. It doesn't affect the signal whatsoever. So we can eject this electroactive species to just serve as a marker of what's coming out of the barrel. We know then everything's working right and so on. Get our cyclic voltammogram out. And then if we know the relative mobilities of acetaminophen, which you'll notice is a neutral marker, so just carried by electroosmotic flow, to, if we know the relative mobility to raclopride, then we can compute how much raclopride we put out. So that's something we're doing a lot now, and these TTL controls help us to be able to do it. And this just gives you an example of an experiment like this. So in this particular experiment, every time there's a little red bar right there, we stimulated dopamine neurons. And you can see every time we do a stimulation, you see dopamine appear. And then we've used some mathematical methods to extrapolate the dopamine out of it, and so that's shown here in the black line. So you see here in the black line, every time we do a stimulation, we get about the same amount of dopamine out, and it falls back to baseline very, very rapidly. And our claim is that this fall back to baseline is due to that process that Andy Ewing was talking about, uptake, the thing that's affected by cocaine, the thing that's inhibited by methylphenidate. So to test whether that's really the case, we took a drug, nomifensine, a drug that is pharmacologically known to be a dopamine uptake inhibitor, and we injected it continuously while we were doing the stimulations. Now you can see that there's a sort of a bunch of stuff going on here. It turns out that nomifensine tends to be slightly electroactive over the potentials that we're scanning to. And this, what looks like an interference here and here, is actually due to the nomifensine injection. But we can use a technique called principal component analysis to extract out the dopamine signal from all the other signals And then when you do that to these data here, you get the gray trace that you see here. So in the presence of nomifensine, much like Andy showed in fruit flies with cocaine, the dopamine signal gets bigger with stimulation, and it falls to baseline more slowly because the transporter is inhibited. And again, it's a very reproducible thing. And in the presence of this uptake inhibitor, you can start seeing little what looks like noise, but those are actually authentic dopamine events. Dopamine is being spontaneously released now in the presence of this uptake inhibitor. You can see that there. Okay, so those are the main features of the data collection. Let's quickly look at what happens in the data analysis section. So you would collect all this data, be on your hard drive, and then you would pick one of those runs, be it continuous, or this is showing you just a single run here. You'd run it in. And again, the key elements of the data are shown here. You'd look at your color plot, you'd look at the current wherever you set the cursors, and you'd look at the cyclic voltammogram by putting that cursor where you want it to be, and you look at the TTL pulses. For data that you collected in the continuous mode that I showed you, we generate what we call snippets. So snippets are taking sections of this data that you have interest in and breaking them up into smaller parts so you can analyze them. So the way you generate the snippets is you pick one of these TTLs and say, I'd like to have all the data for 5 seconds before this white TTL pulse and for 10 seconds afterwards. Something like along that line. So this, by setting up which ones you want to key in on and what kind of timing you want to use, you generate from here a snippet. If you accept that snippet, you hit the accept button, and then you move on down the line to the next white pulse, and you generate another snippet. And this allows you to go consecutively through the data and collect it. What you're looking at here is actually intracranial self-stimulation data. So we were collecting data based on the TTL for the Q on, and there's the Q response, and there's the stimulated response right there. Once you have got a single snippet, then it's probably like you need to filter it. And we used various methods of filtering in the past, but now we're using an FFT method. The FFT method is really nice because more classical methods of filtering data tend to shift it in time. You don't get this shift in with the Fourier transform method, and you get a nice clean signal that you can, with the noise removed. So you could just look at the color plot and see how much better it looks once you've taken the noise out, and you can then do an analysis. Something that's been a real nice feature for our lab is this concept of principal component regression, which Michael Hines started in my lab, and Richard Keithley did an awful lot of it, is now incorporated in our main analysis program. So the problem that solves for us, so here was a stimulation and the pH change afterwards, like I've shown you before. If you plot out what happened at the dopamine potential, you see this thing go down below baseline. And you can clearly see if you look at the color plot, that's because at this potential you don't just have dopamine signals, you have pH signals. So how to separate these apart? We use this method called principal component analysis, in which what you do is build a calibration training set of different cyclic voltammograms. Here you're looking at the one for pH, and you just hit this button, capture standard, and you pick a certain period of time that you want to capture the particular signal for. And then you can do the same thing to capture a bunch of standards for pH changes. And these can be collected either in vivo or in vitro. And then you end up with your cyclic voltammograms here. And the program will give you this score plot that you see here and the so-called K matrix. So what these are as follows. This is a Cook's distance plot here in this plot, and it allows you to look at the principal components and how well they're lining up for each of the components that you're looking at. And in this particular case, they don't line up very well, and the program identifies these 2 points as being the outliers. So what this K-matrix plot lets you do is let you look at what the shape of your cyclic voltammogram is after the principal components have been established. And if you agree with that shape, and that one looks pretty good to me, that's okay, and it would in fact give you a good plot here. But in this case, you see the plot is all sort of funny with crossing of lines here. The pH plot looks pretty ugly, and that's because these points are not lying on the line correctly. If you replace these with points that fit better on the line, you can correct your calibration matrix and get a better, cleaner result. And it's all important to have a good calibration plot. Once you do have a good calibration plot for this principal component analysis, you can do what I think are just remarkable things. So here's some data collected in the brain where there was an electrical stimulation and then a bunch of pH changes. And if you look at what principal component analysis tells you on, it says there's the electrically stimulated dopamine release, but look, under that pH, it was right there, and more dopamine events. Here's the pH change. And then you see another wonderful thing of this approach. The noise comes out of these plots, they're all on the same scale, and is put in the residual. So you put the noise where it's supposed to be. It's not dopamine, it's not pH, it belongs in the residual. In addition, as I was showing you, when you have those snippets and you build them all up, you can use them for signal averaging. And you greatly improve signal-to-noise ratio. So here's some single runs in a behaving animal, and you can imagine a rat shaking his head. You saw him in the video while he's doing that intracranial self-stimulation. So you often need signal averaging to try and get well-resolved signals. Here's the color plot, here's the dopamine signal, and here's the pH signal that you can see very nicely. So anyway, I just wanted to take you through this today and show you all the different things that we have now in this program. And we have made this program available on the web if you want to get it and use it. We actually introduced it last year at Pittcon, but like any new program, it had bugs. We've gone through this year finding the bugs, and they're just about all corrected. And I think by next week, if you go to this website, you will find a corrected version of the program that is relatively bug-free, and where there are problems with it, there's actually an instruction manual. And if you read the instruction manual, you could find out where you can go wrong, okay? So that's kind of good. So let me finish here by just showing you the people who did the work. Ken Brooks is our programmer. Beth Bucher here was the coordinator between our group and the programmer and developed the slides that I showed you today. Nina Owison-White is the one who does the intracranial self-stimulation experiments. This is Annabelle, who's doing the lontophoresis experiments for you. And here we have Susan, who is involved in a lot of the troubleshooting of this program at first. And then involved in our behavioral experiments is Gina Reilley. Thank you very much.
I have time for one or two short questions, and I'm going to take the first question because this drives me crazy looking at Mark's data. It's beautiful, but what I don't understand is how the background current on that carbon electrode stays so constant for up to, 5 or 10 minutes or even longer now?
Conservatively, we've always said about 2 minutes. Now we're seeing it stay stable longer because what I was showing you was big concentration change. So it's drift to signal ratio that's important. But how does it stay stable? So this technique was not developed by myself. It was developed by a guy in the UK in early 80s. He published it in Nature, and I went to the UK to show him, this is total, you can't do this. You can't subtract these big signals. And he had in the lab a digital oscilloscope. This was before computers. He just pushed a button, and there it was. And I saw that, and I said, I'm doing this. This is great. And it just is sufficiently stable.
There's no chemical surface treatment?
We have various, where our limit of our waveform is, affects the stability, and affects stability We've published all those things, but yeah, there is some food.
On your two-dimensional FFT smoothing, which works so well, how did you decide on the cutoff frequencies or the atomization function?
In the temporal domain, we had been using one that I believe had a 6 dB roll-off. So we just took that and went into the time domain and used that same roll-off there. So it's very comparable to what we used in the time dimension, except without the phase shift.
Any other questions for Mark? Let's thank him again for a great talk. Thank you.